Hello all, myself Shruti. Welcome back to my channel. In this week's video, I would like to introduce one more student of BioEd Solutions who carried out biotechnology project. She is Bhavana from Mount Carmel College. She will exhibit her work, research findings, what she has done at BioEd Solutions. So it is on DNA barcoding of few indoor plants and molecular characterization of its symbiotic bacteria. So we all know that air quality is an important factor that plays a role in public health, and indoor air pollution marked with decreased air quality has set the standard very below. So indoor plants absorb carbon dioxide and keep oxygen flowing. We should not plant the plants only for the purpose of decoration or aesthetic sense. It also has lots of benefits on our health. First and foremost, it absorbs lots of carbon dioxide and keeps the fresh oxygen flowing inside the house. They purify the air by removing lots of toxins, and they also help to deter illness, ease tension, and also lower the stress level. It creates a relaxed and happy ambience inside the house. So with all this, it improves one's concentration, boosts up the attention, and also our creativity level will be increased. So there are so many advantages of planting this indoor plants. So first and foremost, as I said, is it improves indoor air quality by removing the harmful substances. So these harmful substances are resulted by lots of our household activities, and this indoor air pollution will cause severe problems like sick building syndromes, chronic obstructive pulmonary diseases, asthma, lung cancer, and it is responsible for lots of deaths in developing countries. So this is one of the very interesting topic where we can overcome indoor air pollution problem by planting more and more of indoor plants. So till now, there is no such criteria or nothing decided which are the plants which helps in removing the harmful substances, but most of the indoor plants helps in removing the volatile organic compounds like formaldehyde, benzene, and trichloroethylene. And these are the chemicals which when present in high levels will lead to lung and airway infections. One more interesting aspect and the significance of indoor plants is it controls the pollution uh, competitively from the aerial elements and also from the root region. That is, the aerial parts of the plants also help in removing the pollutants, as well as the root part of the plants are also helpful in purifying the air, and the maintenance of these plants is very easy. So all of you plant more and more indoor plants in your house and keep your atmosphere very healthy. These plants need not be planted with the soil or pot. If you just keep in the water as hydroponics also, these plants will grow very nicely. So here Bhavna will explain what kind of work she did and how it is beneficial to the society.
Hi everybody, I'm Bhavna. I'm doing MSc Biotechnology from Mount Carmel College. So recently I did my project in BioEd Solution, which is located in Pinia under the guidance of Dr. Shruthi ma'am. The paper was DNA barcoding of few indoor plants and its molecular characterization of symbiotic bacteria. So basically we performed all these experiments in a confined molecular lab which is well equipped with all the instruments necessary for this experiment. So we collected few indoor plants usually available in the garden. Now talking about the indoor plants, as we all know, most of us have at least one or two plants growing in our garden or in the house as well. These plants not only provide like ornamental value, but also they have many health benefits to the human life. Like they accumulate all the carbon dioxide present in the atmosphere and then utilize it and then produce a fresh oxygen, which is basically helping humans to inhale fresh air. And not only this, they also reduce your mental stress. So usually these plants are kept in the houses and in offices as well. So it also increases your memory power, concentration, and whatnot. DNA barcoding is a method of identification of unknown species or using a particular sequence on the DNA. So this method is a promising method which enables all the researchers or further studies on this particular plant. It might be helpful as it contains a lot of information on the. We collected plants from three different families: Asparagaceae, Carassulaceae, and Pandanus. So Dracaena is one of the plant which belongs to Asparagaceae family, where this plant is found in almost 87% of the world. Raceae and Pandanus centenary, these plants not only have their ornamental value but also various health benefits. A person suffering from diuretics or urinary tract infection or talking about the symbiotic bacteria, these are a group of microorganisms present in the root of the plants. So they help in many ecological processes like nutrient acquisition, nitrogen and carbon cycles. These form nodules in the roots which helps in nitrogen fixation, and they play major role in plant development and growth as well, and also protect plants from biotic and abiotic stress. Collected the soil adhering to the roots, and DNA was isolated using the same method followed for the plant, which is the CTAB method. It was mainly done on 16S gene. So, and using the same blast and cluster omega, a phylogenetic tree was constructed, which builds the evolutionary, enables us to know the evolutionary relationship between these bacteria. The data was analyzed using the Sanger sequencing machine and to identify the nucleotides of the bacteria, and then further studies were done. So this further discussions and results will be explained by Dr. Shruthi ma'am, and I thank her for giving me an opportunity to um, participate in her project and help this actually helped me in gaining knowledge about how DNA barcoding is done, and also I got hands-on experience on performing DNA isolation methods and Sanger sequencing, PCR, agarose gel electrophoresis, and not only this, such a. And also, I would like to thank Shruti ma'am for being the guide and help me throughout the experiment and giving me an hands-on experience on all the process which were involved in this. The instruments like centrifuge, PCR machines, agarose gel machine, UV spectrophotometer, giving me this opportunity and also helping me in publishing this.
Two common indoor plants which are present in normally everybody's house, and these DNA barcoding studies of these plants were done. The idea is one thing, she has to learn the hands-on training for this, how barcoding studies will be done. And as I said, these plants aerial parts and roots are equally important. To prove that, we also try to find out the symbiotic bacteria which are present in these plants, and this findings is already published in one of the international journal called Quantum Journal of Medical and Health Sciences. So just to briefly to show what she has done, she has already explained the protocols what she has done. We have just collected few plants belonging to the family Asparagaceae, Crassulaceae and Pandanus. We have isolated DNA from the leaf material, and few genes like nuclear, mitochondrial and chloroplast genes were amplified through PCR technique, and further Sanger sequencing was done to get the sequences of the plants which we have taken, and further bioinformatics studies were done to identify the plants. At the same time, the root samples were collected and the symbiotic bacteria were allowed to grow, providing the favorable conditions, and the bacterial colonies were isolated. They were characterized morphologically, microscopically, and also through molecular characterization. So, coming to the results and discussions, the three plants which we had taken for the study were these three. The leaf materials were taken. The plants were characterized as Dracaena cochinensis. Then the second plant was characterized as Pandanus sanderi, and the third plant is Hylotyphlium eversi. So this is how we did. DNA was isolated, and further, the as said, the genes like ITS2, MADK, PETP, RBCL2, these kind of genes were amplified, and then they were further subjected to Sanger sequencing. With the help of Sanger sequencing, we were able to blast and tell what is our unknown sequence, or the hit which is very near to our unknown sequence. And then phylogenetic trees were constructed to understand the evolutionary relationship of this plant species with the very nearby species. So these are the evolutionary relationship studies what we did. And then, as I said, we isolated two bacteria, and which were identified as Acinobacter calcositis and Pseudomonas sudzeri, which are symbiotic bacteria. So these were also characterized using microscopic and and molecular methods. So with the findings, we were able to tell that the three indoor plants what we had taken, they were barcoded and identified, as I said above. And there is a great um, relationship between the symbiotic bacteria and how the plant helps in purifying the air. So our results also suggest the active interaction of these indoor plants, which helps in reducing the physiological and psychological stress of the person who maintains the plant. And this also helps in suppressing the nervous system activity and diastolic blood pressure, and promoting comfortable, soothe, and natural feelings. So with this, we have at this level we were able to the tell that the plants really help in decreasing the air pollution inside the home. And at the root level, there are few bacterias which we have to explore. And uh, so in the future days, we have plans to find out the role of the plants, exactly what it does to control the pollution. Again, as I usually tell, the idea behind the exhibiting this work is to appreciate and congratulate the students who work at BioEd Solutions, and few students continue their work in publishing their research findings. So it all shows the efforts and also the interest they have towards the subject. So I always encourage this kind of students who come forward to introduce themselves to the research community. They can utilize the company for getting the hands-on experience, and those few students are using this YouTube platform also to exhibit the research findings in the form of video in a very enthusiastic manner. I always encourage such students, and also they can come up with their own project topics so that they can understand and experience the application part of the biotechnology. It gives me immense pleasure to exhibit the students project, and also videos which are made by them, the research findings in the form of publications. And I encourage the students to come forward in the future days also. Thank you all.